Hello, everyone. Welcome to the first MaxWell Webinar. I am Obien from MaxWell Biosystems, and I will be your host for today's session. Thank you very much for tuning in. We are actually broadcasting live from Switzerland right now, and I see that we have attendees from Asia, Europe, US, yeah, many different places. Thank you very much for tuning in, and I hope you enjoy today's seminar. All right. So for today, we will have the following as our agenda. First, very briefly, I will introduce our company, MaxWell Biosystems, and also our technology, microelectrode array. I will also introduce our product, mostly focusing on MAX2, our multi-well high-resolution microelectrode array system. After that, we will have a presentation by our VP for Scientific Affairs, Dr. Michele Fiscella. He will be talking about one of our key applications, which is functional characterization of human iPSC-derived neurons at single-cell resolution. After the talk, we will open the floor for questions. And for today, we will answer 3 questions. And feel free anytime during the webinar to type in your questions through the chat. We will choose 3 out of these. And don't worry if your question is not chosen. So we will contact you directly by email later. All right. So thank you very much again, and let's begin. So we are MaxWell Biosystems. We are based in Switzerland, and we are a team committed to provide electrophysiology systems and solutions to advance scientific discovery and to accelerate the development of novel therapies for brain diseases. Our core expertise include hardware and software for cell-functional assays, and we are experts in using and developing microelectrode arrays. We utilize CMOS technology in our products. So for all of you there, we would be happy to be a partner in your research and in your drug discovery as you find out how your cells work and also what are the effects of compounds on them. Okay, so what is our technology about? Cells, mostly neurons, can be characterized in 2 ways. So on one side, in terms of function, the gold standard is patch clamp, wherein you have a glass micropipette that is placed directly onto the cell in order to be able to measure its activity. On the other side, we can also characterize cells by their morphology, how they grow, and also how they physically connect with each other by using imaging. Our technology sits in between these 2 methods here. So using high-density microelectrode arrays, which is what we offer, we can detect signals similar to patch clamp from individual cells and multiple cells at the same time. And also, we are able to detect the morphology of each cell by detecting the propagation of the action potential along its axons. So this is one example of one cell that is recorded, and we are able to identify the morphology of this cell just based on the activity. This is no label, and you can do this for multiple cells in your sample. Now, focusing more on what we measure. So this is an electrophysiology tool. So comparing with patch clamp, which we have here, for patch clamp, we are recording signals from inside of the cell. So the action potentials look like this, so they are positive. For microelectrode arrays, we have the sensors outside of the cell, so we are recording extracellularly, and the signals look like this. And each one of the spike is an action potential. And for high-density microelectrode arrays, what we have is then multiple of these electrodes at high density. So here we have these electrodes, which are located very close to each other in such a way that we are able to detect signals from a single neuron at multiple points. So here, these are signals originating from along the axon and the soma, and these along the dendrites. So what are the key advantages of high-density microelectrode arrays? So there are MEAs that are low resolution. However, one thing that happens is that if there are cells in between the electrodes, you're not able to detect them. And if there are neurons around each electrode, you're only able to measure the average of the population activity. You cannot know whether there are 3 cells or 5 cells that you are recording from. With high resolution, you can identify the activity of each individual cell. Also, because we have many electrodes at high resolution, we are always at the right spot on where the cells are. Also, we have small electrodes, which can be very close to the source of the signal, so we can get access to cells' small signals such as axonal signals. These are our products. So we have MaxOne, which is a single well microelectrode array system, and we have Max2, which is a multi-well system. For Max2, we have the mainframe, which is where we do the recordings. So this is our data acquisition hardware. You attach your multi-well plate there, and you can also have your recordings for long-term outside of the incubator because this is already a built-in incubator. So the temperature can be controlled and also there is a gas input. Currently, we have 6 and 24 well plates. So for Max2, each of the well actually has the same sensor as Max1. We have 26,400 electrodes within 2 by 4 millimeter square area. And the distance between the two electrodes is 17.5 micrometers center to center. In such a way, we are able to have multiple electrodes detecting signals from a single cell. Another highlight here is that we have low noise readouts, so even the small signals can be detected by these electrodes. Additionally, any of these 26,000 electrodes can be used for electrical stimulation, so you can evoke action potentials in your chosen neurons at any time that you want. All right. So central to both of our systems, Max1 and Max2, is the software, which allows us to visualize and acquire high-quality data, organize the experiments, and perform cell-functional assays. So our software is called MaxLab Live. This is a screenshot of our software. So here you can appreciate the signals coming from our electrodes and detected by our electrodes from the cells. And we have other visualizations such as the electrodes view, which is a 2D view of all of the 26,000 electrodes. And here the red spots correspond to the active cells at that certain point. We also have the raster view in order for you to be able to see some bursting activity or synchronous activity from all of the cells that are being recorded at that time. So here you can see some oscillation occurring in this specific sample. So MaxLab Live features assays or experiments that you can do. Today we are focusing on the neuronal assays that we offer. These are activity scan, network, and axon tracking. Let me go through each one of them. So first is the activity scan assay. So the question that it asks is, are the neurons active? So similar to microscopy, it is able to detect the location of the cells, such as this, but additionally, we are also able to detect the firing rate and the amplitude of the cells, of the signals coming from the cells at that specific point. So here you get an overview of your whole sample, and you can use this to track the development of your culture across multiple days. Next, we have the network connectivity assay. So once we have identified where the cells are, we can continuously record from them. So this is an example of the spiking activity of two different locations. So each spike or action potential is labeled as a circle. And here in the raster plot, one dot corresponds to one action potential. So here we ask the question, are the neurons interconnected? And this assay basically shows us if there are bursting features appearing, meaning the cells are already starting to be interconnected, is there synchrony amongst these cells, and are they really creating this oscillatory activity? And we can also do this across multiple days to track development. So the third one is the axon tracking assay. So from the population, we now move on to the single cells, which is unique to our system. Here we ask, are the axons functional? And here are examples of what you can do. For the same cell, you are able to get the morphology at an earlier date and a later date, compare the amplitude of the spikes of the activity, and also see the elongation of the axonal arbors. Here you can see an action potential propagating, which is detected across one arbor, and we can also calculate the propagation velocity of such an action potential along the axon. So these are the three assays, and they can be applied to any of these neuroscience applications. Of course, our system can also be used for other cell types like muscles, cardiomyocytes, but right now we focus on the neuroscience applications. And these are 2D, 3D, and also retina. For today, we will focus more on the human iPSC-derived neurons, which will be introduced to us by Dr. Michele Fiscella. Michele, the floor is
Hello,
yours.
Marie. Thank you very much for the nice introduction. This is the presentation outline. I will go through the state-of-the-art techniques for assessing neuronal activity, mainly calcium imaging and low-resolution microelectroarrays. I will go through some example studies featuring patient-derived neurons and electrophysiological assays, and I will also show you MaxWell functional and morphological readouts for human iPSC-derived neurons. So the state-of-the-art techniques for assessing neuronal activity. So Marie already mentioned about patch clamp. So there are also techniques that can be, that work at multi-wells, so at high throughput. One of these is calcium imaging. So typically a chemical calcium indicator is added to the cell culture, to the neurons. Upon calcium binding and entry inside the cells, the emission fluorescence change. And typically the image of entire well is summarized by this oscillatory behavior. So from each well, it's measured the width of these bars or the time between the bars, and so on, or the temporal features of this oscillatory behavior. This is a high throughput technique. It can really go up to 1,500 wells. It is also cheap. It measures the average population activity from each well. It is an endpoint measurement, and there are evidences that chemical calcium indicators are toxic, and there are also phototoxicity effects in culture. So low-resolution microelectroarray have been used quite extensively. So typically electrodes of 50 micrometers in diameter and spaced at hundreds of micrometers are used to record neuronal electrical activity. And usually the spikes, the number of spikes, the mean firing rate per well is measured. And even in this case, several readouts focus on the oscillatory behavior on the burst. So again, extracting temporal features from the burst, or the time between the burst, and so on. So these readouts are available at 48, 96 well plate. These are label-free, differently from calcium imaging and long-term, allow multiple readouts per well. They have a very fast sampling rate, so neural waveforms can be extracted. However, they feature low spatial sampling, and single neural readouts are quite difficult. So now I would like to show you how the combination of electrophysiological assays and human iPSC-derived neurons are a very powerful tool for in basic science and also in drug discovery. So, nowadays it's possible, starting from stem cells, either iPS-derived or embryonic stem cell, it's possible with quite different methodologies to obtain neurons and glia cells, and these can be used for pharmacologic studies, for basic science, and so on. The iPS-derived neurons have the advantage that biological samples are closer to human physiology. It's possible to generate these neurons from patients with neurological disorders. They are very predictive preclinical model for drug screenings, and they can also be used for cell therapy to replace damaged cells in vivo. However, they might have phenotype dissimilarity depending on the iPS line. There could be limited in vitro maturation and also differentiation heterogeneity, so different cell types. So for these reasons, very accurate techniques are needed to record eventually small amplitude signals, or also to differentiate cell types and record from the proper cell type in vitro. This is a very interesting study in which cells from patients with bipolar disorders were characterized by patch clamp technique, and it was found that the bipolar cell, the cells, the neurons from patients with bipolar disorders had a higher amplitude threshold for generating action potential. And then again in this study, calcium imaging was used to show that this hyperactivation of these neurons could be reduced by application of lithium. So again, combining electrophysiological assays and the iPS model to study disease mechanism and potentially also drugs that cure the disease. This is another very impactful study in which microelectroarray technology was used to characterize the electrophysiological phenotype of neurons derived from patients with amyotrophic lateral sclerosis. Also in this case, the patient-derived neurons were more active than the control ones, and a drug was used to reduce this hyperactivity, and it is currently in clinical trials. So again, I mean, you can see how the combination of these human neurons derived from patients and the electrophysiological assay is really moving forward neuroscience and drug discovery. So now I would like to show you how our MAX2 functional morphological readouts can be used to characterize human iPSC-derived neurons. So first of all, I would like to thank the laboratory of Professor Andreas Lillemann at ETH Zurich and PhD student Silvia Ronchi, who generated the majority of this data and analyzed this data. This data was presented at the following conferences in US, SFN 2018, ICCR 2019, and all the neurons used are human neurons from Fujifilm Cellular Dynamics International. So here you can see our activity scan assay. You can see as a reference rat primary cortical neurons, very large spikes, can also reach 1 millivolt. So this is the state-of-the-art neurons used in the field with MEA. And here you see human dopaminergic neurons. You see the scale here is 80 microvolts. It's 10 times lower than rat cortical neurons. However, our system enables to record even small amplitude spikes across thousands of cells distributed across the array. And here you see human motor neurons. And the ability to capture the signals from a single cell and to sample a majority of the cells on the array allows to have very reproducible results. Here you see every circle represents a different well, and then you see the mean firing rate of these cultures over time, and you see a very high reproducibility across different wells in the quantification of the mean firing rate. So MAX2 allows for the detection of small amplitude spikes and also for the recording of electrical activity from each neurons in the well. Here, you can also see 4 different wells where we cultured motor neurons. You see how the whole sample electrical activity is very reproducible and similar across different MEA. And now here below, you see the application of reticabine, which is again a potassium channel opener. And here you can see how applying difference concentration decreases and eventually stops the whole network activity. So we can detect pharmacological effect and in, again, a very reproducible way. Now going to our network recordings, here you see raster plots. So each raster plot here with the oscillatory summary population activity is represented on top of each plot. Each one is a different well. And here you see human dopaminergic neurons. And at the bottom, you see human dopaminergic neurons modeling Parkinson's disease. So these are a culture of neurons only. You see the time between the bars is different. However, there was a bit of variability across these different wells. And when co-culturing the neurons with the astrocytes, in both cases, the oscillatory and the amplitude of the oscillation increased significantly in all the wells, and also the reproducibility across wells was very similar. So with the system, it's possible to do studies in which we co-culture neurons with glia cells. We can see the effect of the glia cells on the activity of the cells, and also clearly distinguish the electrical phenotype of different disease lines. Here you can see again the oscillatory behavior, the bars from 4 different cultures, wild-type control motor neurons, neurons modeling ALS, amyotrophic lateral sclerosis, wild-type dopaminergic neurons, and dopaminergic neurons modeling Parkinson's disease. And here you can see that every cell type has a typical burst shape. The burst is very reproducible, and also most importantly, this burst shape is different, very different across different cell lines. So our system enables to detect cell type specific oscillatory patterns. And then, as Maria already pointed out, our system enables to record neurons, their electrical, their extracellular action potential over multiple electrodes. And then, therefore, it is possible to assign each one of these spikes or these extracellular action potential to a given subcellular structure. So typically, the biggest spikes originates from the axonal initial segment, and then the smaller spike propagate along the axons, and it is also possible to distinguish dendritic axons. So questions about the maturity of the neuron, for example, by tracking the axonal initial segment are possible, and it is also possible to study the propagation and the physiology of the axonal signals, and of course also to image the neurons, the neurites of a neurons label 3. So the system allows and gives large-scale access to axon physiology. Here you can see multiple neurons tracked on our platform on MaxWell, where the signal propagate along different axonal branches. And I mean, as an outlook, I would like to show you the quantification of these axonal action potential propagation velocities in wild-type controlled dopaminergic neurons and Parkinson's disease dopaminergic neurons. You see at days in vitro 14, the velocities are very similar, approximately 300, 350 millimeters per second. So in these plots, you see the time that occurs between the action potential in consecutive electrodes and the distance between consecutive electrodes in different neurons. So the data from different neurons is pulled together. And now you see the velocity of these neurons at days in vitro 25, that in the wild type cultures, it approaches values of 500 millimeters per second, which are very close to what we know to be a mature neuron, whereas in the dopaminergic lines, they stayed approximately at 300 millimeters per second, which shows how our system can be used to study, for example, disease like Parkinson's disease, with known to be also an axonopathies that causes the degeneration process in the axon. So in conclusion, I mean, MaxWell enables label-free and long-term monitoring of neuronal activity without interfering with cell physiological processes. It allows for novel and reproducible readouts at both functional and structural level. It also enables to very robustly compare functional phenotypes in human neurons and to measure drug effects in human neurons. And importantly, it enables the study of neural circuits at single neuron and subcellular levels. So, I mean, with this, now I would like to thank you for your kind attention. And of course, if we are now open for questions, we are happy to answer them. Thank you very much.
Yep, thank you very much, Michele, and thank you to everyone for writing in all of your questions. Please continue sending them. I'm just going through these questions now, and many people actually are asking Michele. So I think the first question would be how to culture human iPS-derived neurons on MaxWell plates. What is the coating to use for this case? Yep.
Yes, so I mean, typically the coating and the cell culture procedure that work on regular plastic dishes can be directly used on MaxWell plates. So the coatings that we used are typically polyethylenimine, PI, polyl-ornithine, PLO, and polydilysine, so PDL, as a primary coating to make the surface more hydrophilic. And then, depending on the cell types we used, we recommend laminin or Geltrex or also Matrigel. And different combination of these primary and secondary coatings can be used. And also, again, one can culture only neurons or combination of neurons with glia. And in all of these conditions, we can stably record neural activity for weeks to months times.
Yep, thank you very much. So another question that we have here, also multiple people asked this. So you showed different bursting shapes from different cell lines. Why do you think they show these different features also with and without astrocytes?
Yeah, so I mean, I think that in these cultures, first of all, the ability to really target the electrodes to all of the cells that are present on the MEA and really extracting single neural electrical signals enables to measure these very reproducible shapes. So each neuronal culture, depending whether it was a wild type culture or a disease model culture, will have a different, first of all, connectivity pattern. So the number of neurons that connect to another given number of neurons, and also the propagation velocities of the action potential across this network may vary. So this is why our readouts really enables to extract this cell type specific signature in the bars shapes and in the oscillation behavior.
Yeah, okay, thank you very much. Okay, we are receiving a lot of questions now. For the last one, I think I will go with this one. Okay. So what are the advantages of recording activity at single cell level to characterize a different iPSC lines?
Yeah, so, okay, so first of all, I think across development, what happens is that in the first stages of development, you might have very small amplitude spikes and very few active cells that are active. So first of all, in the initial developmental stage, you can really record, even if 20 cells would be active with this system, with the high resolution, you would be able to detect them. And also in later stages, the ability to really sort out the single neural activity conveys a very high well-to-well reproducibility. So, I mean, we have measured very low coefficient of variation across different wells, which can be directly correlated to the ability to sort out a single neuronal activity.
Okay, thank you very much. Okay, so for all of the rest of the questions, as I mentioned, we will reach out to you by email. And yeah, okay, so that's it. Thank you very much. Please contact us at info@mxwbio.com if you have any other questions or if you would like to request a code or for any other information that you would like to get from us. We are also open to schedule a call directly, so you can have a one-on-one session with any of our application scientists or also with me and Michele with this link, calendly.com/mxw/call. So there you can choose a schedule that is convenient for you, and there will be a 30-minute to 1 hour schedule where we can have these one-on-one calls. We can also have a technical demonstration in these calls. And you can also visit our website for any additional information that you would need. Additional things that I see are coming here are related to like cell densities and also how like protocols to use in order to have to maintain the culture across multiple days. And all of these we can answer to you in a call or also by sending you the protocol by email. Yep. So that's it. Thank you very much, Michele, and thank you very much to everyone who attended the webinar. And stay in touch. We'll let you know what's next in terms of the next episodes of our webinars. And also stay safe wherever you are, and let's keep connected. Okay. Bye for now.
Bye-bye.